Hi there. Thanks for joining today's seminar series. My name is Alex Hoischen. I work in Nijmegen, the Netherlands, and I have a great interest in the effect of rare genetic variants on severe forms of COVID-19. Just to introduce myself and my group that I had here in Nijmegen, my group is called Genomic Technologies and Immunogenomics. So I have a core interest in developing and applying genomic technologies for my own research questions that really focuses usually on rare diseases. But then together with my colleagues, I also try to push latest technologies towards diagnostic use, specifically for optical genome mapping. We're using the Bionano system already little less than 2 years by now and have processed more than 250 samples since then. Next to this more technology-driven interest, I really have a core interest since several years now in immunogenomics, and they're mainly a rare disease focus where we try to study new and novel genes for inborn errors of immunity and primary immunodeficiencies. And again, also here, I collaborate closely with my colleagues in diagnostics, where usually cases that are analyzed for diagnostic exome sequencing with a suspicion for a severe immune defect, my group comes in to reanalyze the data and perform a research follow-up on novel findings. Some of that process that really focuses on exome sequencing here has been described by my previous PhD student in this overview article, if you're interested. This is just to tell you that we already joined the cytogenomics series with 3 other presentations, one by our wonderful collaborator Leila El-Katabi, one by my former postdoc Tuomo Mantere, and I also myself gave a talk already on research findings using optical mapping. The reason why I also joined the COVID-SV consortium is our most recent findings from this summer for very severe forms of COVID-19. So with an entire research team here in Nijmegen, we discovered 2 families in which two brothers each developed extremely severe forms of COVID-19. Sad enough, one of the 2 brothers of family 1 even passed away due to the severity of the disease. And what is truly remarkable about these 2 families is that all 4 brothers are extremely young. They are 31 and 29 years of age in family 1, and even younger, 21 and 23 years of age in family 2. By performing diagnostic rapid clinical exome sequencing, we identified rare loss of function variants in a gene called TLR7, which encodes for the Toll Like Receptor 7. In addition to identifying these mutations that you can see in the cartoon on this slide, we also have shown that the individuals have a defective interferon signaling due to their loss of function mutations in TLR7. And as such, we were able to show that TLR7, once defective, really leads to a downstream defect in mainly type 1 and type 2 interferon. Furthermore, this also implies the TLR7 pathway as an important innate immune response pathway to battle off SARS-CoV-2. Interestingly, our finding was just replicated by an Italian consortium who identified rare TLR7 variants in 6 out of 156 individuals, all male individuals, obviously. And this consortium was also able to show a similarly impaired type 1 interferon defect. This work is also very much in line with a very recent publication by the COVID host genomics consortium led by Jean-Laurent Casanova and an entire team. This group interestingly identified severe loss of function variants in the more extended type 1 interferon pathway and mainly genes that have been previously implicated in severe viral pneumonias. And they found a total of 3.5% of severe COVID-19 cases that potentially could be explained, or their severe predisposition to COVID-19 could be explained by these type of variants. So seemingly type 1 interferon is truly important, and now there's efforts ongoing to really understand what that would potentially mean for treatment of individuals. With this interest in bio-nano optical mapping and an interest in severe forms of COVID-19, I joined the COVID-SV consortium simply because I believe that there must be more rare genetic host factors out there that could lead to severe forms of COVID-19, and they could be very helpful to identify. We may speculate that that again may be related to a functional defect in interferon production. And for those working on more common diseases, I think it's important to reiterate that some of these genetic defects that we identify are individually extremely rare, but collectively they could already explain a certain percentage of cases. Furthermore, these rare disease findings and severe predispositions may allow important insights into the diagnosis, disease mechanisms, and biology in general, and may in some cases even offer novel treatment options. To my understanding so far, there have not been very systematic assessments of structural variants as an important cause or important source of genetic variation. So therefore, I got this interest in the COVID-SV consortium. And I think it's good to point out again that structural variants itself may have a greater a priori chance to be rare and disruptive in the human genome. Furthermore, I think it's important to keep in mind that occasionally point mutations and structural variants affecting the same genes or loci may actually very much complement each other. Especially for extremely rare events, there would be a lot of value in looking at all types of variation. So with that, I hope also the input from my group can add to this SV consortium, and hopefully we can identify novel insights into this devastating pandemic and disease. The work I've been involved in COVID-19 was obviously teamwork from a lot of people here in Nijmegen, mainly driven by my highly talented PhD student, Cas van der Made, heavily supported by my colleagues at Internal Medicine, Frank van der Verdonk, and certainly also my mentors, Han Brunner and Mihai Niteer. We're very much looking forward to see you at the roundtable discussion and answer any questions that remained after this presentation. Thank you very much.